Anatomy of flowering plants, the tissues and the tissue system. Anatomy is the study of internal structure of plants. The basic unit of plants is the cell. Cells organize to form tissue, and in turn the tissues organize to form organs. In a plant, different organs show differences in their internal structure. Within angiosperms, the monocots and dicots are also seen to be anatomically different within angiosperms. To diverse environments, internal structure also show adaptations. The tissues. A group of cells is called a tissue, and these group of cells have a common origin and usually perform a common function. Different kinds of tissues are present in a plant. Based on whether the cells being formed are capable of dividing or not, tissues are classified into two main groups, namely meristematic and permanent tissues. Meristematic tissues. In plants, growth is largely limited to specialized regions of active cell division called meristems. Different kinds of meristems are present in plants. Apical meristem produce primary tissues and they occur at the tips of shoots and roots. At the tip of a root apical meristem is present, while the distantmost region of the stem axis is present the shoot apical meristem. When the leaves are formed and the stem is elongated, some cells are left behind from shoot apical meristem and they constitute the axillary bud. In the axils of leaves, such buds are present and are capable of forming a branch or a flower. Between mature tissues, the meristem is present and is known as intercalary meristem. In grasses and regenerate parts removed by the grazing herbivores, these meristems are present. Both intercalary meristems and apical meristems are primary meristems because they contribute to the formation of the primary plant body and appear early in life of a plant. In the mature regions of roots and shoots of many plants, meristem is present, particularly those which appear later than primary meristem, and those that produce woody axis are called the secondary or lateral meristems. These meristems are cylindrical. Examples of lateral meristems are fascicular vascular cambium, interfascicular cambium, and cork cambium. They are responsible for producing the secondary tissues. In both primary as well as secondary meristems, following divisions of cells, the newly formed cells lose the ability to divide and become structurally and functionally specialized. These kind of cells are termed mature cells or permanent and constitute the permanent tissue. Particular regions of the apical meristem produce dermal tissues, ground tissues, and vascular tissues during the formation of the primary plant body. Permanent tissues. The permanent tissue cells do not generally divide further. All cells of the permanent tissues which are similar in structure and function are called simple tissues. The permanent tissues which have various types of cells are called complex tissues. Simple tissues. Tissues that are made of only one type of cells are called simple tissues. In plants, the various simple tissues are parenchyma, collenchyma, and sclerenchyma. Within organs, the parenchyma forms a major component. The parenchyma cells are generally isodiametric. In shape, they may be spherical, oval, round, polygonal, or elongated. The walls of the parenchyma cells are thin and made up of cellulose. These cells may either have small intercellular spaces or be closely packed. Various functions like photosynthesis, storage, secretions are performed by the parenchyma. In dicotyledonous plants, the collenchyma occurs in layers below the epidermis. It is found either in patches or as a homogeneous layer. Due to a deposition of cellulose, hemicellulose and pectin, it consists of cells which are much thickened at the corners. Cholenchymatous cells may often contain chloroplast and they may be oval, spherical or polygonal in shape. When these cells contain chloroplast, they assimilate food. In cholenchymatous cells, the intercellular spaces are absent. In the growing parts of the plant, such as young stem and petiole of a leaf, they provide mechanical support. Long, narrow cells with thick and lignified cell walls having a few or numerous pits constitutes the sclerenchyma cells. These cells are without protoplast and are usually dead. Sclerenchyma cells, on the basis of variation in form, structure, origin and development, may either be fibrous or scleroids. Generally occurring in groups in various parts of the plant, the fibers are thick-walled, elongated and pointed cells. The sclerites are highly thickened dead cells which are spherical, oval or cylindrical in shape with very narrow cavities. The sclerenchyma cells are commonly found in the fruit walls of nuts, seed coats of legumes, leaves of tea and pulp of fruits like guava, pear and sapota. Mechanical support to organs is provided by sclerenchyma. Complex tissues. The complex tissues work together as a unit and are made up of more than one type of cells. In plants, the complex tissues constitute the xylem and phloem. From roots to the stem and leaves, the xylem functions as a conducting tissue for water and minerals. Mechanical strength to the parts of the plant is also provided by xylem. Four different kinds of elements are composed of xylem, namely tracheids, vessels, xylem fibers and xylem parenchyma. Vessels in xylem are absent in gymnosperms. Elongated or tube-like cells with thick and lignified walls and tapering ends constitute the tracheids. Tracheids are without protoplasm and are dead. The thickenings which vary in form are present in the inner layers of the cell walls. The main water transporting elements in flowering plants are the tracheids and vessels. A long cylindrical tube-like structure, the vessel, is made up of many cells called vessel members, each with a large central cavity and lignified walls. The protoplast in these vessel cells is absent. Through perforations, the vessel members are interconnected in their common walls. One of the characteristic features of angiosperms is the presence of vessels. Highly thickened walls are present in xylem fibers with obliterated central lumen. The fibers may either be septate or aseptate. The cell walls of xylem parenchyma cells are made up of cellulose. The walls of these cells are thin and they are living. In these cells, food materials are stored in the form of starch or fat and other substances like tannins. The ray parenchymatous cells conduct water in the radial direction. Protoxylem and metaxylem are the two types of primary xylem. Protoxylem are the first formed primary xylem elements and metaxylem are the later formed primary xylem elements. In stems, towards the center lies the protoxylem and towards the periphery of the organ lies the metaxylem. Primary xylem of this kind is called endark. In roots, towards the periphery lies the protoxylem and towards the center lies the metaxylem. This kind of an arrangement of the primary xylem is called exarch. Food materials are transported through phloem, usually from leaves to other parts of the plant. In angiosperms, sieve tube elements, companion cells, phloem parenchyma and phloem fibers constitute the phloem. Albuminous cells and sieve cells are present in gymnosperms. Sieve tubes and companion cells are absent in gymnosperms. Associated with the companion cells are the sieve tube elements that are long tube-like structures arranged longitudinally. In a sieve-like manner, their end walls are perforated to form sieve plates. A peripheral cytoplasm and a large vacuole is present in a mature sieve element, but the nucleus is absent. The nucleus of companion cells controls the functions of the sieve tubes. Closely associated with sieve tube elements are the specialized parenchymatous cells called companion cells. Pit fields present between their common longitudinal walls connect the sieve tube elements and companion cells. Pressure gradient in the sieve tubes are maintained with the help of companion cells. Phloem parenchyma cells have dense cytoplasm and nucleus and are made up of elongated tapering cylindrical cells. Their cell walls are composed of cellulose. In phloem parenchyma cells, plasmodesmatal connections exist between the cells through pits in the cell walls. Food material and other substances stored in the phloem parenchyma are resins, latex, and mucilage. In most of the monocotyledons, phloem parenchyma is not present. Bast fibers of phloem fibers are made up of sclerenchymatous cells. In the primary phloem, phloem fibers are generally absent, but in the secondary phloem, phloem fibers are present. These fibers have pointed needle-like apices and are much elongated and unbranched. Phloem fibers have quite a thick cell wall. These fibers lose their protoplasm and become dead at maturity. Commercially, phloem fibers of jute, flax, and hemp are used. Protophloem is a first formed primary phloem which consists of narrow sieve tubes, and metaphloem are the later formed phloem which has bigger sieve tubes. The tissue system. Depending on their location in the plant body, let us now consider how tissues vary. On location, their structure and function would also be dependent. There are three types of tissue systems on the basis of their structure and location. The three types of tissue system are the epidermal tissue system, the ground or fundamental tissue system, and the vascular or conducting tissue system. Epidermal tissue system. The outermost covering of the whole plant body is the epidermal tissue system, which comprises of the epidermal cells, stomata, and the epidermal appendages, the trichomes and hairs. In the primary plant body, the epidermis forms the outermost layer. The epidermis forms a continuous layer which is made up of elongated, compactly arranged cells. Usually, the epidermis is single-layered. The cells of the epidermis are parenchymatous, which contain a small amount of cytoplasm lining the cell wall and a large vacuole. Outside of the epidermis, a waxy thick layer called the cuticle is often covered, which prevents the loss of water. In the roots, cuticle is absent. In the epidermis of leaves, stomata are present. The process of transpiration and gaseous exchange is regulated by the stomata. Two bean-shaped cells known as guard cells are present in each stoma. The guard cells are dumbbell-shaped in grasses. Away from the stomatal pore, the outer walls of guard cells are thin, and towards the stomatal pore, the inner walls are highly thickened. The guard cells possess chloroplasts and regulate the closing and opening of stomata. In the vicinity of the guard cells, sometimes a few epidermal cells become specialized in their shape and size and are known as subsidiary cells. A stomatal apparatus consists of a stomatal aperture, guard cells, and the surrounding subsidiary cells. A number of hairs are borne by the cells of epidermis. The unicellular elongations of the epidermal cells are the root hairs that help absorb water and minerals from the soil. The epidermal hairs on the stem are called trichomes. Usually, the trichomes in the shoot system are multicellular. They may be stiff or soft and branched or unbranched. They may even be secretory. Due to transpiration, the trichomes help in preventing water loss. The ground tissue system. The ground tissue system consists of tissues except epidermis and vascular bundles. Simple tissues such as parenchyma, collenchyma, and sclerenchyma constitute the ground tissue. Usually in the cortex, pericycle, pith, and medullary rays, and in the primary stems and roots, parenchymatous cells are present. The ground tissue in leaves consists of thin-walled chloroplast containing cells called mesophyll. The vascular tissue system. Complex tissues like the phloem and the xylem constitute the vascular system. The vascular bundles consist of both xylem and phloem. Cambium is present between phloem and xylem in dicotyledonous stems. Because of the presence of cambium, such vascular bundles possess the ability to form secondary xylem and phloem tissues and are hence called open vascular bundles. No cambium is present in the vascular bundles of monocotyledons, and since they do not form secondary tissues, they are called as closed vascular bundles. Within a vascular bundle, when xylem and phloem are arranged in an alternate manner on different radii, the arrangement is called radial as seen in roots. The xylem and phloem in conjoined type of vascular bundles are situated at the same radius of vascular bundles as seen commonly in stems and leaves. Usually, the conjoint vascular bundles have the phloem located only on the outer side of xylem. Please share this video with your friends. Leave your comments below and please subscribe to my channel for more content. Click that bell notification that would let you know when a new video is uploaded. Thank you so much for watching. Once again, please subscribe to my channel for more content.